On Tomorrow's World Today, we explore the cutting-edge advances that are shaping 4 different worlds. The world of inspiration, where the wonders of the natural world amaze and inspire us. The world of creation, where ideas come to life from traditional arts. The world of innovation, where ideas and inventions move us all forward. The world of production, where innovations are mass-produced to improve our lives. From Inventionland World Headquarters, here's your host, George Davison.
Ready to jump into a world where tomorrow's tech is today's reality? Welcome to Tomorrow's World Today. Around 5,000 years ago, humans began experimenting with plants and minerals, discovering and testing early drugs, such as willow bark to alleviate headache pain. You know, back then, trying to relieve a headache meant chugging a murky mixture of willow bark. Then in the 1800s, alchemists began conducting experiments, probably because they noticed side effects. They must have been convinced that there had to be a better way than creating mysterious mixtures with no consistent promise that it's going to work for everyone. Now, eventually, they began lab testing to extract pure, safe compounds and developed consistent recipes that could be produced, along with a warning to some people who might have a bad reaction to that medicine. Now, of course, back then, there wasn't some large bank of human data, so the results were at best chaotic. So let's fast forward to today. We have warning labels on the back of all of our prescription bottles. For example, a label may tell us that we should take a medicine with food, or maybe not take it at all if you were pregnant. Lab testing helps to make sure that the medicines that we take are safe today, especially when a specific medicine is going to go out and scale on a national or international level. You've got to go out and collect and test all sorts of samples from around the world in a timely manner, and then analyze all this data to validate this new medicine. Can you imagine the complexity of doing all that? Now, I've heard that a global lab testing network system has been invented, and I'm intrigued, so I'm going to send Greg to the world of innovation to explore it.
At any one time, there are thousands of clinical trials being conducted around the world, resulting in tens of thousands of samples. Now, all of those samples have to be collected, transported, processed, analyzed, and cataloged correctly. And that's why we're here at LabConnect in Johnson City, Tennessee. We're going to meet with Chief Executive Officer Wes Wheeler and his team of experts to discuss innovative solutions to these challenges and more today and in tomorrow's world. Hi, Wes.
Hey, Greg. Welcome to LabConnect.
Thank you very much. I'm super excited to find out about everything that you do here. Let's kind of get an overall idea, though, of what it is that LabConnect is all about. You are a vital part of the drug development process. Tell me about that.
Well, drugs are developed over a period of about 10 to 12 years, and there's a drug discovery portion, there's a drug development portion, and then there's a drug launch, approval and launch. We fit in the middle of that. LabConnect is a provider of central lab services. We work for our clients. Once the patients are recruited, we are able to bring lab kits to the patient's home or to the patient's clinic and take the blood, bring the blood back to the lab. The labs are local, typically. We will test the data from the blood, and then we bring the data back to a central location and deliver it to our clients.
Now, operating labs like that around the country or around the world, that can be a really expensive proposition. How do you handle that?
We have decided, because of the way drug development is done now, patients from around the world are required for clinical trials and for filing. So we have to find labs around the world that are local, because the blood has to find its way to a testing lab within a certain amount of hours, 24 to 48 hours. So we find local labs around the world that are close to the patients. And then we can bring the data back to a central location, back here, for example, collect the data for the delivery to the client.
Okay, great. Well, I'd love to learn more about that.
Yeah, let's talk about that now.
Wes, you mentioned that you have laboratories all around the world. How do you find these labs to work with?
It starts with a protocol. Our client will tell us exactly where the patients have to come from. That varies from protocol to protocol, from trial to trial. So we will aggregate a lot of that information and start to decide what regions of the world we have to have labs in. Over the years, all the trials we've done, we now know the clusters of where the patients typically will come from for a global trial. So we have, based on that data, selected 7 regions of the world, for example, Australia, Europe, of course, North Asia, the US, Latin America. The labs are selected to be close to the patients, close to the clinics, so that we make sure we get those samples back to the lab within 48 hours.
Well, how can you be sure that those labs that you're working with all around the world are following the protocols that LabConnect needs in order to make sure those samples are collected the right way?
No, it's a great question. I think you have to realize every lab is different. They're all owned by different people and different companies. They have different equipment. We have to make sure that the equipment, even if the equipment's not exactly the same, that the test is done the same way, that the assay, which is what we call it, is done in a consistent manner so that the data is collected in a very consistent way, that the parameters, the data output, the data format comes to us in a certain way that we can harmonize the data for our clients.
Well, this seems like a fairly large logistical undertaking, and I know that you have a nimble approach. Tell me more about that.
Patients could be anywhere in the world. We're collecting their blood or their urine or some biospecimen. It could be a tumor cell, it could be a biopsy. That data, that sample has to get to the lab within 48 hours. We have to be nimble enough to be able to be at the location where the sample is being drawn and have the courier collect it, get it to the airline, get it to the aircraft in time. The aircraft has to be on time, and then we have to have a courier on the other end to take it to the lab. That's what we call nimble.
There's a lot of moving parts involved. Now, I know that one of the really important ways that you make sure that the labs are doing things the way LabConnect needs them to be done is with the specialized testing kits that you assemble right here, and I'd really like to get a look at that happening.
I'm going to have Julia tell you about that now. Let's go.
Sounds great. I just met with Chief Executive Officer Wes Wheeler to get an overview of what LabConnect does. Now it's time to get into the nuts and bolts of things with Chief Commercial and Strategy Officer Julia Tarasenko. Hi, Julia.
Hi.
Nice to meet you.
Nice to meet you.
So, Julia, in talking to Wes, I realized that one of the most important parts of making sure that the various labs follow the LabConnect protocols is the assembly of the specialized test kits. And he tells me that you're the person to show me how that's done.
Yeah, absolutely. Specialized test kitting is very important to successful clinical trials. So let me tell you and show you how it's done.
Julia, there are a lot of different components here. How do you determine what goes into a particular test kit?
So test kit is a part of our study design. So the protocol, the individual protocol will determine what kind of kits we use and when. All kits will be custom. They will be custom to a visit, and they will be, in many cases, custom to the patient. Every component will be pre-labeled. So when the site uses them, we know which test belongs to which patient. So I will give you an example. This is one of the tubes we can use. So it's going to be very important how much blood we will draw. Even the color of the tube will be very important and will be protocol specific. So we can have kits that contain 10 components, and we can have kits that contain 50 or even more for highly specialized oncology trials as a part of the personalized medicine development.
Okay, well, I know that the way these kits are then put together is really important to minimize errors and make sure that the data all comes back to LabConnect the way that you want it. So can I get a look at somebody putting one of the kits together?
Yeah, absolutely. Let's have a look. So this is part of the assembly area for our clinical kits. But what we also increasingly do here is what we call companion diagnostic. That kind of kit determines if the patient is eligible to take certain drugs. With the rise of personalized medicine, the drug that's suitable to you might not work on me. So these kits help to determine that eligibility. And here we have April who can help us through the process.
Hi, April.
Hi. So this is an example of our clinical trial kits. Each kit is laid out in the order of the draw for ease of use at the sites.
So when a clinician receives this, they can actually see they go tube by tube right down the line, and then they put it back in the same order. So when it comes into the lab for processing, it's exactly laid out like this.
Exactly.
How many kits does a facility like this produce?
So a facility like this produces about 30,000 kits per month, with well over 300,000 kits a year made globally.
That is really impressive. Well, April, thank you very much. So Julia, I guess at this point, the next thing for me to see is the lab itself, so I can see a kit come back in and head into processing. Can we do that?
Absolutely, follow me.
All right, thank you, April.
Thank you.
So this is our sample processing area, and this is where we prepare our samples before we send them out to our partner labs for subsequent testing. And Dr. Zarin here will walk us through the process of what's happening here.
Dr. Zarin, nice to meet you.
Nice to meet you.
So tell me a little bit about this portion of the facility.
So a lot of the samples that are collected during clinical trials have a stability of roughly 24 to 48 hours. However, sometimes that stability is as low as 6 hours. So here we receive samples within that critical time frame for stability, and we isolate either white blood cells through peripheral blood mononuclear cell extraction, or circulating tumor DNA or genomic DNA, and we extract those isolates and freeze them during that critical time period, where we can then further send those out into third-party labs where they'll analyze those cells and that DNA for further interpretation.
That is really impressive. Thank you very much, Dr. Zarin. Julia, what else do you have for me?
We have sample receiving now, so follow me.
Dr. Zarin, thank you very much.
Take care.
So Julia, we're in the receiving and accession area. And what we're looking at here, this is a part of your new sustainability effort. Tell me about this.
So just to put it in perspective, we receive in a session about 100,000 biospecimens per month. And as an industry, they're all arriving in the styrofoam boxes like this. This helps us to keep an insulation, which means the sample stays within the right temperature. What we have been doing is replacing styrofoam completely with this fully biodegradable and recyclable packaging made entirely out of paper. So if you can see here, the insulation is made entirely of the pressed paper.
And this is going to be as efficient as the styrofoam?
Yeah, absolutely. And we have manufacturing data to support that. And we have taken it one step further. And we tested this packaging in a real world condition, and the results coming back extremely strong.
And basically, at the end of its life, this box will become another box.
Correct. It will be fully recycled and becomes another box.
Julia, thank you very much.
Yeah.
And thank you for spending the time with us.
Of course, my pleasure. Thank you so much.
With Julia, we saw the ins and outs of what it takes to put together a customized lab kit. Now, we're going to learn about logistics and route optimization with Chief Technology Officer Charles Castano. Hey, Charles.
Hi, Greg. Nice to meet you.
Nice to meet you, too. So, Charles, tell me, why is LabConnect a lab like no other?
No, great question, Greg. So LabConnect is transforming the clinical trial space. And the thing that makes us different from any company out there is that we can work across any lab partners worldwide. And what this means is that depending on whether you are in Africa, in Asia, in the US, we can actually take your data, ingest it, and create a data set that you can read through our client portal from that perspective. At the core of this transformation is what we call Lab Connector. And what it does is it allows us to harmonize the information from the data test results that we receive from across the world into consistent data that we can provide to our clients.
So essentially, you're decentralizing the traditional model, but that's unifying you globally.
Yeah, absolutely. But at the same time, I think what's important is that we try as much as possible to be as close as possible to where the patients are. And I think that's the core value of what LabConnect can offer, which is to work with companies everywhere in the world, as close as possible to the patients, so we can do clinical trials at any place in the world.
Well, that seems to present some pretty sizable logistical challenges to work around the globe like that.
There's a lot of challenges, but with our technology that we call sample logistics, with Hawkeye technology, we can address that question. Let me show you how.
Please do.
Yep.
Now we're talking. Well, Charles, I really want to take a deep dive on this technology. Tell me more about Samplegistics.
Absolutely. I'm so excited about our Samplegistics with Hawkeye technology. So what it is, it's actually comprised of 3 different technologies that we put together into one solution. One part of it is about a mobile app that we provide to our site collection partners to make sure that they have a way of inputting information on the sample that they've collected on a mobile device. It allows us to make sure there's no human error in terms of writing. What they can do is take a picture and type in information on a mobile app so that information is provided to us simultaneously. The second one is about RFID. An RFID allows us to actually track exactly where the sample is in the network. So what happens is that that sample travels from a LabConnect site to the site collection, back to LabConnect, and then to a lab partner. The RFID allows us to determine and detect if the sample tube has actually arrived at our facility, as an example, right? And then the third one is courier integration. And what we do is we work with many companies, courier partners out there, to make sure that we're able to gather information from them on notification that allows us to determine if there's a problem with a specific sample or a route from that perspective.
So basically, you're turning your lab partners, everybody at LabConnect, and even your couriers into one system where they're all working together to make sure that the samples show up when they're still viable, because that window is so critical, making sure that it gets to where it's supposed to be.
Exactly, and that's what we're doing.
Excellent. Well, now, as we look to the future and what this technology might bring as we move ahead, everybody talks about AI, and it's a big word that means a lot of different things to a lot of different people. How do you see that being integrated into a system like this?
We do a lot of AI applications and agentic AIs across the company, but let me give an example of its application on Samplegistics. So one example of it is route optimization. And what that means is that, as you know, you and I travel, right? Sometimes there's a problem with weather patterns, or sometimes even like political situations, right? What we're able to do is overlay that information of weather patterns with the courier notifications, and then allows us to create what I call next best action. It provides our team members with information that allows them to determine what's the next best route if a specific route has a problem or issue.
So when it comes to that critical stabilization period where a sample has to get to a particular place in time, this is an aid in doing that.
Absolutely.
And I guess this is one of the things that makes LabConnect
A lab like no other.
Charles, thank you very much. This has been great.
Thank you very much, Greg.
Back at Invention Land, Wes Wheeler, LabConnect CEO, is meeting up with George Davison to discuss lab testing innovations in tomorrow's world.
Hey, Wes, welcome to Invention Land.
George, great to be here. Thanks very much.
Well, I was waiting for you to get here because Greg was telling me all about this great technology you guys are working on. I was hoping you could expand on that for me today.
Yeah, I think of us as a tech company, not just a lab company. So we are a company that doesn't own labs, but we do pretty much everything else required in a clinical trial. So we make the kits, we set up the systems, we collect the blood, we bring the blood back to our partner labs, and then we process that testing data so we can turn that into a technical file for our clients to file for new drug medicines. We also track the movement of the samples from all over the world into our partner labs in real time.
And how are you doing that? How are you tracking all of this through the physical world?
We've found an RFID technology that's working. We're going to put an RFID tag on every tube, and tubes are how we collect the blood. Mm-hmm. So every tube will have an RFID tag, and every time that tag hits a reader, we'll know exactly where it is.
That makes sense.
Yeah.
So, because there's labs all over the world that you're working with, somehow or another you must have invented a system that ties everything together.
So think of us, again, as a tech company, connecting all the dots. So all the systems we have, whether it's collecting data, collecting information, collecting sample locations, locations in the freezers when we store them, all that data will be connected together in one total system. Many different systems, but connected together in one digital format.
Well, you know, Wes, it sounds a little like the technology's moving in the direction of kind of like a rideshare company or a B&B.
Yeah, very much so. So we don't own the labs. We don't need to own the labs. We partner with the labs. We're located next to the labs, but we don't have to own them. It's very similar to a B&B or a rideshare company where they don't own the fleet, they don't own the rooms, but they own the client and they own the data. Very similar to what we're doing. We own the patients in terms of how they're recruited for a clinical trial. And of course, we own the data that comes out of the blood testing that we do with our partner labs.
That makes sense. And so if I'm looking out into the future, I mean, it sounds to me like by tying all this together with this system, you're going to speed up the process of making this happen, which is quite an invention, but won't it also add to the, like all the other pharmaceutical companies that need your services? It's like adding to the invention process itself for new medicines.
Clinical trials are getting a lot more complicated now, so the more that we can digitize and tech enable the movement of blood samples and testing data, the faster we can get those testing data into the file for approval of the new medicine.
Well, thanks for making a better way for all of us.
Thank you for having me today.
Well, everybody, that's another edition of Tomorrow's World Today. We'll see you next time. So how about we check out one of our innovation labs?
We'll do that.
All right. Yeah, we just started building these new labs.
And remember, tomorrow's world is where inspiration and creation drive innovation in production. See you next time.